Yeah, I mean, I guess we'll be talking mainly about your work on neural fingerprinting and naturalistic stimuli or task paradigms. Kind of before we do that, I thought I'd ask a little bit about something you mentioned, you tweeted about a few days ago, which I thought was kind of interesting, and I'd be curious for you to elaborate a little bit on. So you said, just got reviews back on a grant. It's still in funding limbo, but one reviewer wrote such beautifully supportive comments of me and my work that I'm tearing up at my desk. I don't know who this reviewer is, but if you're reading this, thank you. This encouragement means so much. So yeah, I'm just curious if you could expand a little bit on that. That's not an opinion you see people tweet about that much.
Yeah, it was a rare more personal, it's still work related, but sort of a rare personal tweet for me. Yeah, I submitted a grant last summer, as one does, and waited, 6 long months to hear any news. And, you finally get this email and it's like, well, we're feeling positive, but we still don't know if we can fund it. And so start reading. There were 5 reviews, and you start reading through them. And, everyone has constructive criticism, as they do. So some nice comments too, but there was sort of one paragraph in particular that stood out where one of the reviewers just wrote this long paragraph just saying, that he thought that me personally and the type of work that I was doing was really novel and innovative and, was really pushing the field forward. And he sort of said, I'm going to go on to provide constructive comments because that's what we do. But to be very clear, I have a very high level of enthusiasm for this investigator and this proposal. And I don't think she needs to, revise it and wait another year. I think you should just give her the money because this is a really cool direction. And she has this proven track record. And yeah, as someone who's only been in this PI role for a few years and you never quite know how people perceive you and how your work is landing. And obviously I've been fortunate to have some papers that I think have made an impact, but I don't know, just sort of getting that kind of personalized encouragement from clearly someone who I'm assuming is a fairly senior person in the field, totally anonymously, right? So they're not getting any credit necessarily for that from me or anyone else. Other than this tweet, I suppose. And so, yeah, it was just sort of, it really moved me, to be honest, because, yeah, we work very hard on these grant proposals and most are not funded and sometimes you get lucky, but just to even get that piece of encouragement was really lovely.
Did that change a little bit how you're going to write grant proposal reviews yourself?
I hope so. I hope that I will be in a position to pay it forward. I've sat on a couple of grant panels, not too many so far, but I think it was a really nice reminder of, when you really like someone's work, tell them, like it really makes a difference. And I try to do that and I have done that in the past, but I think this recent experience was a good reminder and a good push of, how much an impact that can make on someone, because our field is filled with a lot of rejection and waiting and just, kind of hard news. And so, yeah, I hope that I can continue to pay that forward.
Yeah. I guess to get to your work and kind of what you do and what you've done so far, I thought we could just take the kind of something that I do very frequently, which is a kind of biographical path of getting there. I'm curious, you had an article in the New York Times about you studying linguistics that I didn't read because I don't have access to the New York Times, but I was just curious. I think I could see the first 2 lines where it says something you liked studying foreign languages or something like that. I'm curious, why did you decide to study linguistics?
Yeah.
Just briefly, how did you get to write an article in the New York Times about how you studied linguistics? That's slightly random.
Well, maybe I'll start with earlier than the article and kind of work my way up to the article. So growing up, I'm pretty sure the first sentence of that article is growing up, I wasn't a science person or something like that. Maybe not. I don't know. You saw the first sentence.
It was something about how people in your, somewhere early on, there was something about people in your school wanting to be like lawyers or something, or like going to finance or something like that.
Yeah. So, I grew up, I went to a good public high school, and I was surrounded by, lots of professionals. But in high school, I was always very interested in languages and learning foreign languages. I kind of started with French, and then my school opened up a new German program. So I took a couple of years of German. I was kind of teaching myself Spanish on the side. I was just, I really loved languages. which was not super typical in my high school, but, there were good enough classes, so I was able to explore that. And then on my college applications, I didn't know exactly what I wanted to major in. I kind of just wanted to take a smorgasbord of languages. I didn't even necessarily want to commit to one single language. So on my applications, I remember marking linguistics as my intended major, which of course here in the US, it's infinitely flexible and you can switch majors. It wasn't like I was committing myself at the application stage to that, but I was indicating my academic interests. And to me, the most natural thing was linguistics because it seemed like it sort of gave me this opportunity to study some actual languages, but then also to learn more about how language works, on a more fundamental level. And so I ended up going to Yale for my undergrad, and I did take some linguistics classes early on that kind of confirmed that, yes, I was interested in this subject matter. I did take one class called Language and Mind, which was sort of the cognitive science and a little bit of the neuroscience of language as well. And that really, really piqued my interest. And then I ended up from there taking the sort of intro to neuroscience class that was offered at Yale at the time. And that was sort of half to fill a science requirement. So again, here in the US, you have to kind of take a wide variety of courses at many of these liberal arts schools. So I kind of needed a science class, but then I also kind of had an inkling from this other linguistics class that I might be interested in the brain. And I really, really loved that class. And I kind of considered double majoring between linguistics and neurobiology, but I knew I never thought I wanted to go to medical school. So, and the neurobiology major at the time was kind of more of a pre-medical. There were many classes that you had to take that sort of overlapped with the pre-med requirements, which I was less interested in. And so I ended up just sticking with the linguistics major, but I added a lot of coursework in neuroscience. And I ended up doing a senior thesis in the linguistics department, but it was with a professor who was doing some fMRI of language. So this was an undergraduate senior thesis, but I was very lucky in that this professor didn't have any graduate students at the time. So I was actually able to kind of start to take on the role of almost more like a graduate student with really helping to design and run this study. It was an fMRI study of these kind of very esoteric, like long syntactic clause sentences that, are not something anyone would ever choose to utter, but nevertheless could be understood by people. And so we were sort of looking at kind of pronoun movement and syntactic resolution of clauses in this sort of esoteric way. But at the time, I loved it.
I guess your interest in German must have helped a little bit there. Because
yeah,
we're certainly
in English these sentences are like horrible, but I suppose
German is right.
They're fairly common. Yeah. So I was doing this thesis my senior year. I was kind of sinking all of the free time I had into thinking about the study, running the study, reading literature. And then in parallel, I was figuring out what I wanted to do next after graduation. So I was watching all my friends, prepare their med school applications or go through the finance or consulting interviews. And I just really had no idea what I wanted to do. And I was kind of throwing a bunch of applications at different parts of of the world. Basically, I was like looking at international fellowships. I was looking at some nonprofit jobs. I was just kind of struggling to find anything that would feel like a fit. And, in the meantime, I was like really loving my research. And I remember having a conversation with my undergrad advisor in the early spring of my senior year when many of my friends had already kind of figured out what they were going to do. And I still didn't have any acceptances or any really much to show for any of my efforts. And she was kind of like, well, you really love this research. Like, why don't you just go to grad school? And it had never occurred to me, I'm sort of embarrassed to say, even after 4 years of college, that I could actually, become an academic and do this type of thing for a living, right? And I think, as I said, like growing up, I was surrounded by lots of professional people that had wonderful careers, but I didn't know any professors or academics growing up. I think even after, again, making it through 4 years of college, I kind of had a pretty limited view of what professors actually did. So I hadn't really thought about a PhD, but that comment really prompted me to introspect a lot and say, yeah, this research is the thing that I'm really drawn to, that I actually enjoy the most. And so actually, that's when I wrote the New York Times essay. I think the New York Times had put out a call that spring for, essays from college students just about what was on their minds and how they were picturing and planning their lives after graduation and stuff like that. So I drafted that and sent it in, and it was very cool that they published it. But yeah, the upshot of the essay was sort of this research kind of snuck up on me, or this career snuck up on me, in the sense that it was kind of something I had been doing, but I hadn't quite put it together in my mind that I could turn this into a career. From there, it was not necessarily a straight path from there. So I ended up sticking around for one more semester after graduation to wrap up the research, or in theory, to wrap up the research I had been doing for my thesis. As often happens in science, the honeymoon period kind of ended. So I think we got a bit lucky with the first couple of studies or analyses that we did for my thesis, and things were working really well. And I was, super excited with all the discoveries we were making. And then of course, our follow-up studies were a lot less clean, and things got messy. And I kind of saw the side of science where it's hard, because you're working really hard and you're not getting clear results, and things aren't working the way you expect them to. And so I had put in a couple of applications for graduate programs that year. So I guess the fall after graduation, I was working as an RA and I was applying to grad school. But kind of even by the time I submitted the applications, I wasn't totally ready to jump into a PhD program right away. I think I was realizing that, yes, I love this stuff, but maybe I should explore some other things before I fully commit to this journey.
Mean? Like you weren't sure about it, or you thought there was something you needed to learn before, or?
I think I was feeling that, this thing that I had really loved, like maybe I only loved it because it had been going well. Like maybe I just got lucky. And then once things started to get difficult, I was kind of like, oh, wow, okay, this is hard. Like, it's just emotionally hard. You feel like you're working really hard, and things aren't working out the way you expect or hope. And it's not clear if you'll have anything to show for your efforts. And so I was kind of going through that and seeing that side of things. And it made me realize, like, okay, it's not just like you show up to work every day and make a discovery, and then you show up the next day and make another discovery. It's like these are long, winding, difficult roads. And I mean, that didn't totally scare me or put me off, but I think it did make me realize that I needed to be really sure that this was the path I wanted to take in order to commit to at least, 5 to 6 years of the PhD, and then potentially beyond that if I were to stay in academia. So I didn't think it was fair to either myself or my potential mentor, for me to start a program if I was experiencing this kind of uncertainty about whether I was genuinely ready to kind of throw myself into this.
Would you recommend that to your, you have undergraduate students now who probably ask you that kind of question? Because, I mean, in some sense, it seems to me it probably is a good idea to commit to the PhD period, but after that, I guess it's, you're not committing your life to, you're not signing off to a life in academia necessarily. But I'm just curious, like, would you, because I guess many people aren't sure whether to do this or not, right? And in many ways, you only find out by doing it, I guess. But I'm just curious, like, whether your perspective has changed on that a little bit, or just what you'd tell people.
So I often give the advice that I've never met anyone who took time off and regretted it. I have met people who didn't take time off and regret not taking time off, right? I've also met people that were really sure what they wanted to do and just went straight through, and they were fine and they thrived. But I've also met people who went straight through because they weren't quite sure what else they would do, or because of this sense of, these training periods are so long and I want to get wet. It doesn't have to be PhD either. I mean, it could be medical school, it could be law school, or, whatever the next step you're considering. I think, especially when you come out of these kind of prestigious pressure cooker schools, there's this sense of like, okay, what are you doing? And, you see all your friends like going on to the next big thing. And it can be scary to kind of take a pivot and do something for a couple of years that is not what you want to do long term, but it just kind of gives you that breathing room to figure it out. So I mean, I often give advice, whether solicited or unsolicited, to undergrads or other people that are kind of at a crossroads of like, if you do end up going on to a PhD, let's say, I mean, no one ever said, or I've not met anyone that said at the age of 40 or 50, like, oh, I wish I got my PhD 2 years earlier. I mean, yes, it's a long road, but it doesn't really matter. And like at the end of the day, it doesn't really matter if you take those extra couple of years in terms of your overall life timeline, but it does matter in the sense of if you take those couple of years and it increases your confidence that this is actually what you want to do, And that can kind of help sustain you through the difficult times of like, well, I tried a couple other things, or, I did take that time to kind of explore and I came back to this. It's kind of like that silly old saying, like, if you love something, let it go. If it comes back, it's yours. It's like, if you do actually circle back to this, I think that's very meaningful. And yeah, as I said, I've never really met anyone that took a few years off and regretted that, but I have met people who did not take a little time off when they had the opportunity, and then later on said, what, yeah, it may not have changed their ultimate path, but it might have just given them that confidence or those extra experiences to kind of power through when things do get tough.
Yeah, it's interesting that you said that no one's regretted that, because I guess I've sometimes voluntarily, sometimes not so voluntarily, taken time off between each step. I went after school immediately into my undergraduate, but then I had it kind of just happened that I did a 1 year working as a research assistant. I guess in Europe, masters and PhD are separate anyway, usually. So I had a master's, but then a summer job, they offered me to stay on longer, and it's like, why not? And yeah, I definitely don't regret having stayed there and then done a bit of that. And then after my master's, yeah, it's funny because it was very similar in the sense that I think we were a very good year with very good people, most of whom went on to do PhDs at very good places. And I remember when we were applying, I just felt this, I don't know whether the others also felt it, but there's this sense if you don't figure out what you're going to do and where you're going to do PhD in the next few months, that's basically it. Obviously, if we thought about it, we knew it wasn't, but it felt kind of like you had to figure it out this time. And then I ended up starting a PhD and then quit that after a few months because it just wasn't the right thing, even though it was supposedly on paper a very good PhD. And the funny thing is, quitting a PhD is obviously also quite different from just taking time in between, because then there's the question for future PhD supervisors: why did he quit? Is he going to do it again? That kind of stuff. But even now, though, I'm going to start a postdoc in a few months. First of all, no one even knows I took a I'll tell them, I don't mind, but obviously I'm sharing this right now in the recording, but no one knows about it, no one cares. A lot of people said they wish they'd quit, actually. they just waited too long. But yeah, it's interesting because, yeah, as I said, I've taken some pauses, also a few months now after my PhD, I'm taking a few months pauses. There's definitely nothing to regret about this few months here and there, or a year, or in one case even 2 years of not doing that. I'm a little bit older now, but I don't care.
Yeah, totally. Yeah, I think that's most people's experience. Yeah, so I guess to pick back up that thread, I'm realizing I'm, proselytizing about taking some years off, and I didn't even get to what I actually did. So I was applying that cycle sort of right after I graduated, and even by the time I sort of pressed submit on the applications, I was waffling a little bit as to whether I was fully ready to jump in. And so I ended up moving to Peru for about a year and a half. As you do. I got a job working for a coffee company. This was really random. But I hadn't studied abroad in college, which was a bit ironic given my love of and interest in languages. And so living abroad was something that I really wanted to do and experience. And so what was supposed to be kind of just a 6 month, I think I was telling everybody, including myself, that I'll just go for 6 months, basically in between applying to grad school and starting grad school. But I got down there and it was great, and I was having a blast. And I did actually get into a couple of places that year, but it just became clear to me that I wasn't ready to commit. And so I tried to actually defer my acceptance from Yale, because I was pretty sure that I wanted to go back to Yale for my PhD, and they wouldn't let me defer. And I was already in Peru, and I just said, I wrote some very heartfelt emails, but I was just like, I'm not ready. And so I'm going to decline, and thank you so much. I'm so honored for this opportunity, but I don't think it's the right move for me or anyone else, for me to start a program right now. And so I stayed in Peru for about a year and a half. Had some really fantastic experiences, and then I ended up kind of starting to miss science, right? So I had interest in kind of international development and actually journalism as well, and I was able to explore a little bit of the development side in Peru. But I did start to miss science, and I never knew, I never thought I was going to be there forever. So I started to think, well, maybe I want to explore science journalism. Maybe I want to do science communication. And so when I first moved back to the US, I was working at MIT in their news office, and I was writing kind of press releases or kind of public facing articles for their homepage. And that was really fun. So I got to interview, a bunch of amazing scientists at MIT when they would have big papers come out. Our office would do a big press release, an article on it. And so I got to kind of flex those muscles of, taking interesting scientific findings and trying to communicate them to a lay audience. And I always like to write. And so that was really fun. Like I had a lot of fun in that role, I think. But then part of it ended up being kind of this gateway drug back into science itself, because at some point I realized, like, I don't want to be just writing about science. I want to be the one doing the science. And especially when I would interview people in their BCS, their brain and cognitive sciences program, about their work, I just would want to like stay in those conversations, just keep talking about what was happening. And that kind of showed me like, okay, maybe I am ready, maybe, and nothing I had been doing, even though the work in Peru was really interesting, and then the work at MIT was also quite interesting, like nothing that I was doing sort of on a day-to-day basis in either of those roles really captured my attention or passion or interest the same way that research had. And so that was a good sign that, okay, I've sort of explored these counterfactuals, so to speak. I've kind of tried these other careers that I thought might appeal to me, and I liked them, but I didn't love them. I didn't love them as much as the research that I've been doing. And so that gave me the confidence that maybe I was ready to go back and commit to the PhD. I don't think at that time I was necessarily thought I was committing to academia for the rest of my life, but it was, even just a 5 or 6 year, here in the US, we don't often do masters. Some people do, but it's possible to go straight into a PhD, and then it takes 5 or 6 years, and that's a chunk of time. So, you want to be sure that you're ready, even just for that step. But I did feel ready at that point. And so I reapplied just to Yale. I was lucky that they took me back. Todd Constable took me back, and kind of the rest is history, I guess.
Yeah. Just a kind of last question about the time kind of before you started, I guess, doing what you're doing now. Yeah, how did the thing with Peru happen? I mean, like, why Peru? How does one? I mean, did you have a specific language or country or region or? And then, I mean, you said you worked for a coffee company, which is pretty generic. I'm not exactly sure what that's supposed to mean. So what did you actually do when you were there, and kind of why that job? Or
I wanted to go somewhere Spanish speaking. I never formally studied Spanish, but it's a very common and wide-spoken language, both here in the US and all over the world. So it was a language that I wanted to learn and kind of cement for myself. So I wanted to be ideally somewhere in Central or South America. And the opportunity was, I think found it like on a pretty random, there used to be a website called idealist.org, which was kind of nonprofit jobs and stuff. So the company that I worked for was an interesting one in that it was technically a for-profit coffee company headquartered in the US in Portland, Oregon, but it was one of these kind of social enterprise models where they worked very closely with coffee farmers in coffee growing countries to improve the quality of their product, which can vastly increase the price that they can sell it for in these markets in the US and Europe. So the headquarters, again, was in Portland, Oregon, but they had these supply side offices, one in Mexico, one in Lima, and one in Tanzania. And so the model was to kind of apply for grants or pour some of their own profits back into helping coffee farmers improve their equipment and their practices to kind of get the highest quality coffee, which you can then sell for, many, many times the price of, a typical batch of coffee just on like the New York Stock Exchange or whatever. So it was a very interesting business model. I mean, actually, what I was actually doing for them was a lot of communications and grant writing, and but I was based in their Lima office, and I got to travel a bit to the coffee farms in Peru, and it was really fun and really interesting. I can't say I use a lot of that knowledge at this point, other than I drink a lot of coffee to fuel my neuroscience habit. But so that was it.
Okay, it sounds fun. Especially if you're into coffee.
The ironic thing is I did not actually drink coffee before I worked for that company, but now I'm I'm pretty addicted. And I wish I could say I have like great taste in coffee, because this company was very much about like high quality coffee and getting the best possible beans from, from the trees into your cup, and every step along that process like can have an effect on the final taste of the coffee. In reality, I just drink whatever's. I mean, I can appreciate good coffee, but I'm not a snob. I just need any coffee. That's fine.
Maybe that's. I also don't drink coffee. I just never like the taste of it. Maybe that's how I can get into it too. Go to Peru and work there for a few years. Anyway, we've kind of reached the point where you start doing research in a more full-time, more committed sense. Yeah, you said that Todd Constable let you in again, or something like that, I think was your phrase. So was it clear from the beginning that you were going to work with him, or and if so, why?
I think it was clear to me in the sense that I think I'd had enough experience by that point to realize that who your mentor is and who your advisor is has a big impact on, not only what's happening in your PhD, but also beyond. And so I'd gotten to know Todd a bit in undergrad, because even though my primary advisor was in the linguistics department, we were running an fMRI study, and it was her first fMRI study at Yale. So we were relying very heavily on Todd, who was at the time and still is kind of the director of MRI research at Yale. And so I got to know him quite well through that experience and other members of his lab. And yeah, I think at that point I was ready to commit to the PhD, but I really wanted to know that it was going to be an environment that I could work well in and be happy in, and with people that I could work well with. And that was kind of a known quantity for me. So I didn't explore too heavily beyond that environment. And yeah, so
and he didn't mind you deferring and then coming back again?
Yeah, I mean, I think that was another benefit in retrospect, because now as a PI, I mean, I'm sort of, I encourage people to take non-traditional paths. I'm very open to students that have gone and done stuff outside, but now sort of looking at the path to PhD, at least here in the US, it's like, usually you do a bunch of research in undergrad, and then you spend 2 years working as a lab manager or a research assistant in another lab, and then you apply to grad school, and so you have sort of these experiences under your belt. And I wasn't coming in with that full-time RA experience right after college. And so I guess that made me a bit non-traditional, but I guess Todd was willing to take a chance on me, so to speak, because he'd seen me or he'd worked with me when I was doing my undergraduate thesis. And that was kind of evidence enough for him that, I, if I was coming back, that I was serious and, I was the real deal and I could get stuff done. So yeah, I'm not even sure anyone else would have even looked at my application, given that I'd sort of taken this weird detour. Maybe they would have, but it just seemed like a good fit.
I mean, that's the question, right? Because in many ways, it's also just way more interesting someone who does something unusual and isn't too afraid to, I mean, in a sense, like, I'd rather have someone, I mean, I'm not a PI, so who knows what would happen if I were a PI, if my opinion would change. But I feel like I'd rather like to have people who are a bit interesting and not just went straight down the path that was laid in front of them. I mean, it's fine if you do that, but...
I agree with you, but I also recognize that it is a bit of a risk, right? Taking one of those students. People have varying levels of openness to that, but Todd, I think, was secure enough at the time and kind of had some data on me, so to speak, from before, and was willing to give me this opportunity. So.
Okay. And then I don't actually know what your... should probably look this up. What your initial projects were? What was it? Wasn't language again, right?
Actually, funnily enough, my very first project in grad school was on dyslexia, which is not something I had studied necessarily in my linguistics major in undergrad, but obviously language related. And so Todd had an existing data set in collaboration with some dyslexia researchers, the Shaywitzes at Yale, very well known dyslexia researchers, and they had scanned something like 140 kids and adolescents with dyslexia. So my very first project was looking at differences in functional connectivity between typical readers and readers with dyslexia. And that was good. I mean, it was an interesting project. We had some interesting findings. I think it served to kind of help me get my feet wet with the functional connectivity methods at the time, which was what Todd's lab was interested in, and ended up forming the bulk of my PhD. So that was my very first project. And then from there, we kind of moved into the more individual differences and behavioral prediction, fingerprinting stuff.
Yeah, I wanted to, I guess we can just move straight to the fingerprinting stuff now. Yeah, to kind of, in a sense, introduce the topic. Um, I mean, I guess no one likes justifying their research, but I am going to ask a little bit like what the why to do that. And so first, you I think you summarized this very well later in a review in Neuroimage. You wrote, but why do we care about individual differences in neuroimaging measures in the first place? Most researchers are probably not interested in brain-based fingerprinting for its own sake. After all, there are better ways to identify someone than going to the trouble to scan them and calculate a brain connectivity profile, e.g. DNA, actual fingerprints, simply looking at or speaking to them. And to that, I'd maybe add... So I don't actually know whether this is true, but it seems to me that kind of the uniqueness of a human fingerprint is kind of just a random occurrence. I don't know whether there's any like meaningful, but I don't think there's any meaning. I think it's kind of just... It just kind of happens to be that way, and it happens that is very useful in some criminological circumstances. So kind of what is the kind of specific application of neuro fingerprinting? That's kind of... I hope you kind of comment a little bit on that too. Yeah, so we know why we're even talking about this in the first place.
This is a great question. And maybe to answer this, I'll kind of back up a step and say that the fingerprinting result in that paper, so the 2015 paper really has two sides to it. The first side is we can fingerprint or quote unquote identify someone based on their brain connectivity across different days or across different scan types, et cetera. So that's sort of a brain to brain matching. You know, we're matching one person's brain in one session to the same person's brain in a different session. The other half of that paper is the behavioral prediction side. So can we actually take that brain data and use it to predict something about behavior outside of the scanner? So that's sort of brain to behavior as opposed to just brain to brain. The fingerprinting side of that paper was very much a happy accident. So it was not the result. I mean, this, you know, when we tell our post-hoc story about it, this is not the story we tell in talks, but this is the real story. So we, at the time, so it was myself and Shilin Shen, who is a fantastic research scientist in Taz Lab at the time. We were actually co-first authors on that paper. And we had gotten excited about kind of dynamic functional connectivity and also differences in functional connectivity between brain states. So, for example, between rest or performing different tasks, which was popular at the time. And so we were playing around with data from Human Connectome Project, and we had calculated these sort of sliding window connectivity matrices, and we can get into what that means. But essentially, we were sort of taking snapshots from different individuals. And in that data set, the Human Connectome Project, there's many individuals who were all scanned while doing different things. So each individual had 2 resting state scans and then several different task scans, like, for example, a working memory end-back task, an emotional faces task, relational matching task. And so we had these snapshots, so to speak, of each individual under these different conditions. And what Shilin and I first set out to do was say, okay, if we have a bunch of these functional connectivity matrices calculated from different tasks from different people, and we throw those into a clustering algorithm. For example, can we cluster connectivity based on task state? Can we see differences, reliable differences between what the brain looks like as it's doing an end-back task versus, you know, doing an emotional faces task based on the whole brain functional connectivity patterns? And so, again, we had this sort of set of matrices coming from different individuals under different conditions, and every time we would throw them into these clustering algorithms, we kept getting out subject instead of state. So all of the matrices from the same subject would cluster together. So like your matrix from rest would look more like your matrix from end-back than my matrix from rest, for example. And so, like, as it often happens, at first, you know, we're sort of showing these results and everyone's like, well, that can't be right. I mean, because the usual model in fMRI is, you know, you give a task and you think that that's kind of your manipulation of brain activity, right? Like, brain activity changes in responses to tasks. Like, we know this. This is why fMRI works. So why aren't we seeing, you know, consistent task patterns? Why do we keep getting subject out? But then that result was so strong and we couldn't kill it. And so we ended up just kind of leaning into that result. And that ended up being the finding, which was that actually there's a lot of stability within subjects and also uniqueness across subjects to these whole brain functional connectivity patterns, such that you always kind of look most like yourself, regardless of what you're doing. And yes, tasks do change and modulate the connectivity on top of that, but they don't do so much as to make me look like you, for example, or vice versa. There's some sort of stable backbone that's always present in my brain that, yes, can be modulated slightly depending on what I'm doing, but it's strong enough to kind of identify me across these different contexts or days. It wasn't the result that we set out. I mean, it's not what we hypothesized, but it's what we found. And so I think it was a nice example of kind of following the data and letting the data tell us, what the most important thing was. And then we started to kind of formalize that angle of things and kind of poke that in different ways and see if we could make an interesting point about that. And it's funny because, yeah, I mean, as the quote that you just read kind of highlighted, I mean, to me, it always felt like a bit of a party trick of like, oh, look, we can identify people using functional connectivity. And we thought it was a compelling point, or we thought it would be compelling for fMRI researchers in particular, right? Because there's a lot of kind of the traditional dogma of fMRI is like, yes, it works, but it's pretty noisy. And therefore you need to aggregate data over multiple individuals to kind of find the effect that you're looking for, right? And this was kind of turning that on its head a bit and saying, actually, there's a lot of individual differences that are, that they can be somewhat noisy, but they're stable enough within a person that you can actually look at, this individual person's data. And that is the signal in that is sort of strong and consistent enough across different sessions or different brain states to uniquely identify that person. And so we thought that that would be compelling for the fMRI community. We didn't necessarily expect it to be as compelling as it was to like a more general audience because of exactly this point that you just read out, which is kind of, well, we don't need a method of identifying people based on fMRI. It's more like this is a methodological point we can make about, there's enough stability and enough uniqueness in these signals that they could potentially be meaningful. And so for me, the more compelling piece back then and still now is, what do these fingerprints, so to speak, what kind of information do they actually carry about people? Because the analogy that I like to give, which I think picks up on what you were just saying, is, it could be the case that these functional connectivity profiles are just like barcodes, right? Like if you go to a store and you are looking at objects, like each one has a unique barcode that identifies it, but there's nothing in the pattern of thick or thin stripes, for that particular object that tells you anything meaningful about the object. Like if you were just to see that barcode in isolation, it wouldn't index anything meaningful about what that object is. That could have been the case with these fingerprints. We were hoping that it wasn't, right? We were hoping that there was some meaningful signal in there that related to behavior or the actual real world output of the brain, right? And so the second half of that paper was trying to prove to ourselves and others that, there is meaning in these things. And so that's where we ended up showing, at least in that first paper, that you can use resting state connectivity patterns to predict, to some degree, not a perfect degree, but with some degree of statistical accuracy, predict people's levels of fluid intelligence based on a test that they had done outside of the scanner. And so that was kind of the piece that I think we needed, again, to prove to ourselves and others that these were not just sort of, in fact, that kind of makes the fingerprint metaphor break down, right? I mean, the fingerprint metaphor is useful to kind of quickly convey what it is we're doing, but I fully agree with you that, the actual pattern of bumps and ridges and grooves on people's fingers generally is, not, maybe there's some pseudoscience that analyzes fingerprint patterns, but I think generally it's accepted that that doesn't actually index anything meaningful about a person. And that's in contrast to brain functional connectivity fingerprints, which we do have a fair amount of evidence now, like can be used to index meaningful things about people.
There probably would be, right? I mean, if people do like palm readings, I guess the fingerprint is just too small to really see. So I guess
I'm sure there's, I'm sure there's a community that does it. Maybe, yeah.
Yeah. I don't know how relevant this question exactly is, but it just occurred to me. So I thought I'd ask it. Does this have any ethical implications for data sharing? I mean, in a way, sharing fMRI data, in a way, it feels to me it wouldn't, because I guess you're already sharing like lots of information about people. But I'm just curious, like whether that has thrown up any kind of particular implications there for you, or in how you share your data and that kind of stuff.
It's funny because when the fingerprinting paper first came out, we did get a fair amount of media attention, which, I was a grad student at the time, and it seemed really fun in the beginning. And then it was actually quite stressful because we would do these interviews with journalists who would actually, be sort of pressing us to admit that this has some privacy concern. And I don't want to entirely dismiss that. I mean, I think, any amount of medical data, you have to think about privacy. You're still much better able to identify someone based on their anatomical, like T1 scan, for example. And so when we share those, we often deface them, and there's kind of ways around that. And of course, anytime you're sharing data, you want it to be very much anonymized and de-identified, and there's no way to link it back to the individual. I mean, in theory, right, with functional connectivity fingerprinting, if you had a scan from someone and you knew their identity, and then even if there's other scans of theirs kind of floating out there that have been de-identified, you could potentially link it back to this scan. I mean, I think the extent to which that poses a privacy concern really kind of depends on where we're going with this. Like, is it actually possible to get, I mean, I sort of said this before. I mean, we can get some degree of prediction accuracy for this fluid intelligence metric, but it's nowhere near ready to be rolled out in like a clinical or real world setting. You know what I mean? And so I think for me, to some extent, it's like, well, how much of a concern this poses really depends on how good our models will get, because it's hard to imagine a world where, I mean, unless you could meaningfully extract additional information from someone's scan, just kind of knowing like, okay, this scan came from this person's brain. And again, I don't want to dismiss it, because I think many people are working towards that, and that's sort of like our ultimate goal or hope, is that, these things will have real world relevance. But I think right now, I mean, there's also the compliance piece of like, you have to agree to an MRI scan and lie actually quite still and be very compliant for a decent chunk of time in order to even get high quality enough data to do this, right? So it's not like we can just pick up on fingerprints that people have left behind on objects unwittingly. I mean, this is something that people, volunteer for these studies, and then we're quite careful with how we anonymize and de-identify the data. So again, I don't want to entirely poo-poo it. I think it's a thing that we have to be thinking about, but I think ultimately, if the goal is to use these scans to develop, real world tools that are useful, then hopefully we can kind of do that in a way that respects people's privacy, but also, leads to some kind of meaningful clinical tool that could be helpful. So, yeah, I don't know.
I mean, I guess the point is that the main thing you're able to, I mean, for example, as you said, like you can identify people across tasks, but it's already in the data that this is the same person, right? So it's, you're not really from that immediate case, you're not gaining that much. One thing that just occurred to me, can you know how like often you have the same participants coming over and over again at like certain scanners to take part? I'm just curious, do you think you could, let's say you take like the fMRI papers that came out of one institute and just figure out like, actually you only have like, you say you tested like 1,000 people, but actually was only like 300.
Interesting. Yeah. I mean, maybe. I mean, again, it's like, it's hard to be fully sure, because like most things, like this is not perfect. And even when we're getting like 90% accuracy, there's still going to be some mistakes that we make. So I think it's hard to like conclusively prove. You could say, oh, there's a chance, my confidence interval is this only comes from, half the subjects that's saying it does. But yeah, I think it'd be hard to like fully.
Yeah. And it's not like you would invalidate the data or anything like that. I was just curious, like whether you could, because I guess that would be like a way to use the general approach with existing data to see kind of how far it gets you, all that kind of stuff. But yeah, so maybe kind of the obvious question. So what does identify people? Is it just some very nuanced statistical correlations between brain areas that are just, they're in the data, but as a human, you can't really, in that sense, do much with it? Or is it kind of fairly obvious that like, I think you mentioned, I think I saw a talk of yours online, for example, that the social areas or something like that, or social stimuli are particularly good for identifying people. So I'm just curious, I guess we're going to move also more towards naturalistic stimuli and that kind of stuff now. Yeah, what in or decreases how easy it is to fingerprint someone neurally?
Yeah, it's a good question. So in that first paper, we did a couple of analyses where we kind of broke things down by brain network. And so we were asking, are there particular regions or networks of the brain that contribute more to this identification, that are basically sort of like more distinguishing of people? And I want to be careful with how I present this result, because we did find something fairly logical, which is that the higher order association cortices, so things like frontoparietal area, frontoparietal network or default mode network, things like that, which had been known, from a lot of prior work to have more variability, both in terms of their anatomy and their function across people. Those were the same regions that, relatively speaking, did better at identifying people. That being said, one thing I think is also really important to keep in mind is that, identification was possible well above chance based on all of the networks that we tested, so like it might be a difference between like 85 and 90% accuracy, for example. So yeah, you can say like, there's some regions or networks that carry slightly more of this information, but I do think the information is very widely distributed in the connectome. And there's been some really interesting follow-up papers, not by us, but by folks like Dan Kennedy and other groups that have shown that even if you just sort of select a random set of edges or connections, maybe as few as like even just 40 connections, I think they found in one paper, that are just randomly selected from all over the brain, like that's enough to fingerprint people with a decent degree of accuracy. So I do think that there are particular brain networks and also states, which we can get to next maybe, but that do like a bit better, for example, than like average, and that's important to pay attention to. But I think it's also important to emphasize that the information is very widely distributed, and you can get very good fingerprinting even with only like a very small slice of the functional connectome, so to speak, that's kind of randomly drawn from all over the brain.
Does that tell you anything interesting? Like, I mean, in a way, I would imagine that, for example, something, like the brainstem or motor cortex, something like the things that have to connect to things outside of the brain very directly, or like early or sensory cortices. I would imagine that those might be a bit more stereotypical in people, and that the more abstract you get, the more it's down to just random chance of how things, work out in a particular brain. I'm just curious, like, is there anything there that tells you anything kind of fundamentally interesting kind of about how brains are structured, or is it just kind of what I said, it's the kind of fairly obvious things?
I mean, I think the fact that even things like motor cortex or subcortical brainstem areas, they don't do as well, again, as the higher order cortices usually, but they still do quite well, right, in terms of fingerprinting. And so I think, part of that could be due to perhaps less functionally interesting reasons. For example, one question that we got on that paper, and we still get, which is a great question, is, how much of this is just anatomical differences between people that then when you do your registration of your data and then you're applying this sort of group level atlas, although you can also do this on individual level, like how much of that is just sort of anatomy, which we know is slightly different between people, right? And so then that's going to have an impact on your functional signals if you're kind of grouping them according to these anatomical regions that are not exactly the same in everybody. So I do think that differences in anatomy contribute to this signal for sure. And we know that anatomy differs across individuals, even in these kind of lower order regions, so to speak, or kind of typically more stereotyped regions. But I don't think that's the whole story, again, because we do see differences across brain state in how well we're able to identify people. And anatomy is not changing across state, at least not at those short timescales. So I think there's additive components of both anatomy and function that are at play. And I think the fact that this information is so distributed, again, like probably some of it is due to anatomical differences that are present all over the brain that you can always pick up on. But I think, even in some of these systems that we do think of as more stereotyped and more consistent, I mean, there's still individual variability in those systems too, right? And so I think it's a combination of all of those things.
Yeah. I mean, you'd imagine that anatomical differences would play a fairly major role, but
they definitely contribute. But again, I mean, and we did a couple of things to try to rule out that as the only reason. We definitely, I think that is part of the reason, but to me, the most compelling evidence against that being the only reason for these differences is the fact that we do see sometimes pretty substantial differences in either how well we're able to fingerprint people or how well we're able to predict behavior based on just what the person is doing during the scan, right? And anatomy is not changing within the course of a scan, between a resting state run and a movie run, for example. So the function is definitely adding something on top of that that's meaningful for the the individual signals that we can get out.
Yeah, so let's then move towards different kinds of paradigms you might use and how they're changing. You had a paper called, I believe, Is It Time to Put Rest to Rest? In which you talk about 3 waves of, types of stimuli or tasks that people do when in a scanner. So maybe just as an introduction, kind of what are the 3 waves, and then we can get into a bit more detail.
Sure. So in that paper, I was conceptualizing of this as, okay, so the first wave was, if we go all the way back to the early 90s when fMRI first came on the scene, people were using, I think rightfully so, pretty simple and standardized tasks where they had a very clear handle on the timing of the task. So, you're asking someone to tap their finger at a particular time, and you know when that time is, and therefore you can search in the motor cortex for activity that increases as a function of tapping your finger, right? And then there's visual tasks, which are very well controlled, where you're presenting flashing checkerboards or other stimuli to try to map out retinotopy, for example, and you have very discrete trials, and you know exactly what the person is seeing and when, and the stimuli are simple enough that you can model them appropriately, and that was sort of the whole general linear model or GLM approach, which was the bread and butter of the early days of fMRI, which again makes a lot of sense. It's a brand new tool. You want to make sure that you're doing studies in a way that you have clear hypotheses and you can validate the methods and understand and trust the signals that you're getting from this new bold contrast, right? And then, so that was kind of the first wave, was these well-controlled, very carefully designed tasks. Then the field moved into what I called in that paper the second wave, which was resting state, right? So one of the first papers was Bharat Biswal's paper in 2005. It took a few more years than that to really kind of catch on. But I would say like by the early 2010s, people were really really getting interested in these spontaneous fluctuations, which could be measured while people did nothing at all and just sort of lay in the scanner. Well, they're doing something. We just don't know what, right? So people are doing something, but it's not necessarily being controlled by the experimenter. But we're able to measure, we get these really high dimensional signals, and we're able to look at spontaneous fluctuations and use those to understand something about how the brain is functionally organized. One of the reasons people first even trusted resting state scans in the first place is because a lot of the co-fluctuations recapitulated what we were seeing during tasks, right? So the left and right motor cortex, for example, tend to be active together even while people are resting. And that kind of mirrors, what we see in tasks, or the frontoparietal network, for example, which we had seen during classic working memory tasks. Both the frontal and the parietal parts of that network would sort of be active often when people were doing working memory. And we can see the same thing, those two discontiguous regions are also active together when people are just resting. So that was kind of the second wave of, well, maybe we don't need this tightly controlled task structure, and we can just get a lot of information from looking at the dynamics of those signals as they arise more spontaneously. And I think that was great. I think that was extremely productive. Obviously the proof is in the pudding. There's been a lot of advances that we've made, I think, in our understanding of brain functional organization just from observing the brain at rest. But the argument I was making in that paper was, I personally think it's time to take a lot of that knowledge and a lot of that creativity that we were spurred to by this resting state, because resting state, it really puts a lot of the burden on the analysis, right? Like, you have no experimental design, and so all of the creativity and all of kind of the intellectual burden is on, well, how are you going to analyze the data in a way that allows you to get something out of it and draw insights that are interesting and interpretable. And so we gained a lot from that, for sure. But my argument is that, so now we can sort of take that as inspiration and move into this third wave, where we're not necessarily just having people lie in the scanner with no idea of what they're doing. And we're also not going all the way back to the more simple control tasks, although I do think, there's still a place for both of those things. But we're now at a place where we can really move forward into this third wave, which is thinking about how are we going to come back to experimental design, where we do have more of a handle on what people are doing or might be doing, and design new paradigms or come up with clever new ways of taking the new analysis approaches that we that we developed in the second wave of resting state, and taking those into a place where now we can start to link some of those fluctuations to ongoing cognition or sort of ongoing thought processes, which we don't have a great handle on at rest. And I would argue we also don't have a great handle on when we're using very simple artificial feeling control tasks. So it's kind of a marrying of the task-based approach, not going necessarily all the way back to the simple controlled tasks, but thinking about kind of a new wave of tasks that are closer to what the brain might be doing in everyday life, but that we as the experimenters still have some knowledge of and some control over. So there's some ground truth as to what people might be feeling or experiencing in that moment. And then can we relate that to these very high dimensional signals that we're recording using fMRI, taking some of the analysis approaches that were inspired by the foray into rest.
Mm-hmm. So just before I forget it, why is the term naturalistic stimuli not ideal?
This is a soapbox that I've tried to get on, and I think it's not. I still use the term for the record, but it's funny because well, no, I mean, I think I like to bring up that point to remind people. So, and just to define it, I guess, for folks. So, like usually when we say naturalistic stimuli, we mean things like watching movies or listening to stories in the scanner. These are often stimuli that were not expressly designed for research purposes. So these are, movies that were made for the purpose of entertainment or education or something else, or podcasts that are, entertainment or instruction. And so these weren't crafted for scientific research purposes. And nevertheless, they can be very fruitful to use in neuroscience and psychology experiments. But I think the idea that a movie is naturalistic, I mean, a movie is very deliberately crafted and very just deliberate and specific choices about editing and script. And it's funny, like people often think of, like, for example, luminance, so just the brightness of the movie at any given time is kind of this low level confound, right? Like you want to regress that out. But if you talk to a filmmaker, they're like, luminance is not, I mean, luminance is a very deliberate choice. You make choices about the lighting depending on the mood that you want to set. And so I think it's just important to remember that, it's not like we walk down the street and experience our lives as a Hollywood movie, right? I mean, there's a lot of editing and crafting that go into these stimuli that are designed to really, have an effect on people. And that can make them very powerful stimuli. But I don't think, I think it's a mistake to think that, if we were to walk around with a GoPro on our heads, that's maybe the most naturalistic movie stimulus, but that's not the one people use in the scanner. And again, I don't think that's a problem. And I think we can capitalize on these stimuli that are really powerful, that are really engaging and make us feel things and make us think things in ways that maybe just GoPro footage from a street wouldn't. So it's not an argument to not use them. I mean, quite the contrary. I think it's just, if we think of them as naturalistic, we're sort of doing them a disservice in the sense that they are very deliberate artistic products, and we should think carefully about what they were originally intended to do to their audience in order to assess, if and whether and which of them is the most appropriate stimulus for our particular use case.
Yeah, this might not be 100% directly relevant to it's relevant to the whole conversation. I recently had an interview with Nachum Ulanovsky, who does really cool stuff with, he does spatial navigation in bats. So recording, individual neurons as bats are flying around and doing all sorts of stuff. And we talked a little bit about, for example, he, says that the whole natural versus controlled, it's not like one dimension, it's two dimensions. And you can find these behaviors that are highly controlled in the sense that animals do them naturally and repeatedly, and that kind of stuff. And that, it's not necessarily a compromise you have to make. It's more of a two-dimensional space on which all kind of behavior fits. Again, it wasn't exactly directly related to what you said.
Yeah, no, that's I think it's interesting to think of it as not just a unidimensional continuum, but a more multidimensional space.
How do you get the films, for example? How do you choose them? Do you there's millions of films out there, not counting everything you can find on YouTube or TikTok or whatever. How do you select the right stimuli to be, I guess, in that sweet spot between, I don't know whether it needs to be in the sweet spot between kind of a natural video and controlled, but kind of how do you, there's so many to choose from, how do you select the right one?
Yeah, it's a good question and a hard question. And the irony is that I'm actually not much of a movie person, or like I I have terrible taste in movies and I like haven't seen very many of them. And so my students are always like making fun of me uh, because I never have good suggestions. But yeah, I think it's a tough question. I mean, I've been sort of on this soapbox for a few years now of like, maybe let's replace at least some of the resting state acquisitions with movie acquisitions, because I think that you get more bang for your buck. They're more powerfully and flexibly analyzed. I think having that ground truth time course of what's actually on the screen or in people's ears at any given moment gives you a really nice handle into what's actually going on with the brain activity. But of course, then the question becomes, well, which movie should we use? And it's a tough one. And I think we're still in sort of early days of this work. And I would never pretend that there's sort of like one optimal stimulus or even like an optimal set of stimuli. I mean, the cop-out answer is it very much depends on the research question. But, I think, for example, like, so my lab is very interested in individual differences in how people interpret the same stimulus. And so one thing we've done is we've searched for sort of more independent films that people are less likely to have seen before. And we've searched for films that do kind of leave open a little bit of ambiguity as to what might be happening. It's actually harder than you might think. So I spent a non-trivial part of my postdoc, don't tell Peter, oh, I think he would have been supportive, like literally watching indie films on YouTube and other websites to try to sort of like find stimuli that were doing what I wanted them to do, in the sense of creating a story world that was interesting enough to be engaging, but still ambiguous enough to be plausibly interpreted in different ways by different people. It turns out to be harder to find those than you might think. I think most directors and filmmakers are quite good at what they do, and they always have kind of some interpretation that they want you to get out of it. And so even if it feels ambiguous, there's often sort of less variability in what people ultimately come to than you might want there to be, at least with these professional and semi-professionally produced films. So that kind of leaves you with a couple options. You can make your own films, which is difficult, although perhaps with generative AI getting less difficult. Yeah, I mean, I think you want to ideally, I think, select stimuli that are going to be engaging for people, number one. I mean, I think that's part of the benefit of using naturalistic stimuli, is that it's actually more enjoyable for subjects than simply lying there and resting, or usually even performing a more traditional task. And I think it is important to think carefully about the time varying features of the movie. So I talked about, luminance before being this like confound, so to speak, but you can actually pull out these feature time courses of not only low level things like luminance and loudness, but also like mid-level object categories, like, is there a face on the screen? Is there a house on the screen? You have language, if you have a movie with dialogue in it, and then you have these kind of high-level features like, what is the emotional tone, or is there a suspenseful moment and thing? And usually for those, you have to get kind of human raters to annotate them, and people may or may not agree on those things. But I think one practical piece of advice that I've given folks is, when you're planning a movie study, like if there's something that you're interested in, like maybe it is the language or the dialogue, or maybe it's kind of that emotional tone, you can kind of try to pull out a lot of these features and just see to what extent they co-vary across the film. If you're really interested in studying these high-level emotional changes, and you don't want that to be confounded by lighting changes, for example, like you might want to look at that beforehand and kind of see, okay, if those two things are very strongly collinear, where like every time the movie enters this like dark phase, it literally gets darker, then you can make your argument that maybe that's not the best stimulus, and you want to kind of be able to separate kind of these lower level things from these higher level features. So that's just sort of a very like practical piece of advice that I've given folks. But I really think that at the end of the day, we have enough evidence that having any movie on board for many, not all, but many use cases is better than just resting state, that you can't really go too wrong. And there's not a super principled way, I would say, at this particular moment to choose a film. And so go with what you think is going to resonate with your participants, whether they're healthy college students or children or older adults. Ideally, something people haven't seen before, because that can introduce another element of variability if some of your subjects have seen it, or if you're taking a clip from a longer film where some people have seen the whole film, that could change kind of if some people are able to contextualize that clip in the context of a larger story. So there's sort of these experimental variables that you might want to think about. I have a paper with a former postdoc, Claire Grawl, where we kind of outline some of these practical considerations. But I would say also just don't be afraid to pick something that you like and you think is compelling and you think is going to resonate with your audience, because that's really, at the end of the day, like what we're using these for is to get people engaged and to kind of drive their brain in these interesting ways.
I suppose it shouldn't be too emotional that people get too many movement artifacts. I could imagine, especially a comedy could lead to a lot of like head movement because of people laughing. I don't know whether you've had that, but
yeah, I guess we haven't used stimuli that are like so funny that people are actually laughing in the scanner. It's interesting. So there's been some work showing that, especially in harder to scan populations like kids or patients, movies actually overall decrease head motion relative to rest, at least. The other big question that we've grappled with, I don't think anyone's published a comprehensive treatment of this, but is whether head motion is correlated across people, because that might not be ideal if you want to use some of these intersubject correlation approaches. So like if you and I are watching the same movie, do we actually move our heads at the same time, right? Because maybe there's some scary thing that jumped out, or maybe we're both laughing. And we've looked into this. We haven't published anything on it. It usually tends to be less correlated than you might fear across people. But yeah, that's definitely something, or like physiological signals, if everyone's kind of like holding their breath. I mean, I think there's lots of things that you might want to try to measure and record. But in general, it seems like for most populations, the data quality during movie watching is just as good, if not better, than at rest. And that's in terms of things like head motion or arousal. People are more likely to stay awake, I think, during movies, and physiological artifacts and stuff like that. So that's been our experience so far.
Another kind of pretty big and obvious question, at least to me, is I think most people think that the more natural and normal, the more everyday kind of stuff people have to do and experience in a scanner, the better. But like, you have to be able to analyze it. And what am I going to do with people watching whatever film for 20 minutes, an hour? I don't know. Probably depends. I suppose you could just repeat the stuff that's been developed for resting state. I assume you can pretty much just apply that more or less one-to-one, but I'm curious, like, what's the, kind of what do you do with all that? Okay, so now I have data of people watching film. What do I do with that? I mean, you already mentioned you might have to get people to rate certain events or all that kind of stuff. I'm just curious, like, what do you do with the data?
So I think this is one of the coolest things about naturalistic data is they're very flexibly analyzed. So the same acquisition, the same scan, you can really analyze it in lots of different ways. And so you can do all of the same functional connectivity analyses that you might be able to do from resting state, where you actually just sort of forget about the stimulus altogether, but you're kind of analyzing these fluctuations or the sort of intrinsic organization of the brain. You can compare that across different movies if you want to, or you sort of like use the stimulus in some sense in that way, but you can just ignore it and do your sort of classic bread and butter functional connectivity analyses on movie data. But perhaps more interestingly, or as more of a point in favor of actually going to the trouble to use movies, so you can, I was sort of alluding to this before, but you can actually extract features from a movie and model them in a very similar way to how you would do it for a more simple task. So movies do carry visual and auditory information that can be modeled. I mean, you're not controlling it, but you can model it, so you can get kind of a luminance time course for how bright the movie is at different moments, and you can take that as a regressor basically back to your brain data, and hopefully you see some changes in visual cortex, for example. If you're looking at luminance, you can do the same thing for auditory information. You can model these more mid-level features and recover, for example, like fusiform as faces are on screen or in response to the changes in faces when faces are appearing or disappearing. You can treat movies like a task in the sense that you can model different features of the task and see which brain areas care about those features. Again, you have less control over the features, so some of them might be collinear, and so you might run into problems that way. But the benefit is that you're getting this really rich multimodal. I guess if you have audio and visual information, you're getting multimodal, but then you're also just sort of getting the brain in this much more naturalistic, I guess, for lack of a better word, but you're not using these very stripped down stimuli. You're sort of giving the brain input that's closer to what it might be experiencing in everyday life, in the sense that there's a lot of stuff on screen all at once. You're kind of paying attention to different things. And so you can kind of use movies to confirm findings that people might have discovered in more controlled stimuli, to say okay, once I now build up into this screen that's messy, and there's a face over here and there's a house over here, and there's movement and the luminance is changing all the time, do I still see some of the same regions that I might expect that cared about these isolated features and these lower level tasks are those same areas caring about those same features once I sort of build out to this more complicated situation. And then you can also potentially discover new relationships between features and brain areas, or like the interaction between different features that might not have been possible with the lower level tasks. So that type of approach really, I think, requires you to have a forward model of the movie, so to speak. So you have to know kind of what features you want to model, and you also have to know how to model those features, right? And that's maybe pretty straightforward in the sense of like, is there a face on screen or not? But it becomes more difficult in the sense of these higher level features of like emotional valence time courses, or like suspense or anxiety or things like that. And so it's very possible. And so you can get human annotations of those things. And so that's one thing you can do. You can also take these, I think of as more data-driven intersubject approaches. So this idea of intersubject correlation that was first introduced by Uri Hassan back in 2004, and I think is an extremely powerful and elegant way of looking at the data. So in that approach, you're saying, I don't really know what it is about the movie that the brain cares about, and I'm not going to assume that I can model it, but I am going to assume that there's some correspondence such that if you and I watch the same movie and it's, time locked in the same way, that there's probably some shared processing going on, such that if I take the same region in my brain and correlate that activity time course with, that same region in your brain, if I see that you and I are correlated in our activity as we're watching this movie, I can infer that that region must be processing the movie in some way, right? Because otherwise, why would it show a correlation in its activity across two different brains at two different times and two different scans, et cetera? And so you can kind of use one person's brain as a model for another person's brain rather than modeling the movie directly. You're saying, okay, the movie is inducing some signature of activity that's going to be consistent across brains. And so if I just look for what's consistent across brains, I can start to get an idea of which regions are responding to the movie. And so you can do that both in terms of space and in terms of time. So you can look for particular moments where people become synchronized. You can look for particular brain regions where people tend to be synchronized over the whole movie. And you can use that to try to understand something about group level processing. And you can also take that to an individual difference framework and ask, for example, like, are these two people more synchronized as they're watching the movie than these other two people? And if so, what do those people share that these people don't? And then there's, lots of newer approaches too, where you can look at brain state changes and like event segmentation. So as you're kind of in one part of the film and then you kind of switch to another part, where do you see kind of state changes in the activity patterns in the brain? Can you automatically infer those? You can take an encoding model approach where you're trying to learn mappings between the features of the stimulus and what that does to the brain, and actually predict, in a brand new stimulus, given a subject and the stimulus, what would the different region time courses look like as a function of the known features of that stimulus. And that kind of gives you a handle on, if you can sort of discover the model that transfers stimulus features to brain activity, then you've learned something about, what the brain is actually responding to in the stimulus in this more naturalistic setting.
Mm-hmm. I have in a way, very simple question about repetitions. So I guess one reason why the original task-based experiments are so unrealistic is because you need lots of repetitions to actually get to the effect. How do you deal with that if you have some visual stuff? I can clearly see how you can have lots of repetitions of certain visual features, be it seeing a face or something like that. But when it comes to, let's say, emotions or something like that, let's say you're interested in betrayal, so you show someone a film where someone is betrayed. There's probably going to be one moment where that really happens in the film, right? And not 100. I'm assuming you're not showing them the same 10-second clip over and over again. I'm curious. I would imagine that maybe with naturalistic stimuli, the effect is larger. People actually feel the betrayal more than maybe they might in some sort of random task. But yeah, I'm curious, kind of how do you get good statistical power when you, in my assumption at least, don't have hundreds or at least tens of repetitions?
Yeah, it's a good question. And I think it hits on this there's sort of a more general, I suppose, I won't say disadvantage. I'll say maybe caveat or thing we need to still work out is what actually happens in the brain as we watch the same movie a second time, right? There's sort of this paradox actually in like film studies and literature and other like media studies is like, we do actually often just in our everyday lives, we seek out the same media over and over, right? Like there's maybe like a favorite movie that you have and you may have seen it, many, many times, or like sometimes we return to the same book at different points in our lives and stuff like that. And so like, even though we know what's going to happen, there's still some attraction or enjoyment that we get out of experiencing that piece of media a second or a third or so on number of times. I think it's still sort of an open question, like what happens, what the effect of repetition of a movie clip is, for example, in the brain. Like I think you've never really watched the same movie twice, right? The second time around, you have memory and anticipation effects, or maybe you're actually paying attention to slightly different things. And this is something that people have started to look at this in terms of what is sort of reliable across different presentations and what actually changes as a function of how many times you've seen that stimulus. I think we have a little ways to go to sort of understand that better. I think it just sort of impacts the conclusions you can draw. So in your example of like, if I'm interested in the neural response to betrayal and I have only sort of one film clip and I show everybody that clip and I kind of see what happens at that particular moment where you learn that the character has been betrayed. I think there have been, we've done some of these and there's been other studies showing that you can get interesting information from just sort of that single trial, so to speak, when you're sort of looking across subjects. But I think in that case, you wouldn't necessarily be able to claim that would generalize to any instance of betrayal. So like, I think one kind of happy medium that we've been thinking about, I acknowledge that this is challenging, but I think it's doable, is like, if you can build out sort of a suite of clips or a set of clips or something that all kind of have some moment that feels similar in some like abstract sense. It's almost like you want to abstract away from the details of the individual clip. But if you can get that betrayal moment across, like, let's say 5 stimuli that are all different in terms of exactly what's happening and who the characters are, but then you can have sort of slightly more of a trial-like structure, but you're moving away from that exact particular clip and you're trying to generalize across different experiences. I think something like this would also be useful for longitudinal designs. If you want to scan the same people across development or across some other, probably like a treatment for a mental illness or something like that. Like maybe the ideal thing is not to have the exact same clip. Maybe you want to have one identical clip for sort of test, retest, reliability, et cetera. But then it would be nice if we could move to a model where we have if we have a good parameterization of these stimuli and what's actually happening and what we care about. Like in theory, we could either find or generate sets of stimuli that are all sort of different, like in an item-wise sense, but that all kind of tap into a similar process. And then we're kind of generalizing at that second order, more abstract level rather than the exact like audiovisual details of that particular clip at that particular moment. I don't know if that makes sense, but
yeah, it does. But in a way, I guess one problem maybe is that again, maybe this isn't a problem. It just seems to me like that is that in these artificial tasks, I guess the reason, not the reason artificial, but it doesn't take that long, maybe, until you can experience a broad array of different emotions. You might feel included or excluded or something like that. Whereas in a film, it just takes time, right? It unfolds, at least it feels to me, often more over time. It seems to me like if you have like some very simple like game you made up, you can maybe get like every few seconds, like a little bit of it. Whereas in a film, it might take 5 minutes before you even really understand what the relationship is between the people that you're seeing. I'm just curious whether that just inherently limits how many repetitions you can do of something, or is it just, or am I just thinking maybe also just from the entirely wrong perspective about this?
No, I think you're right. Like, I wouldn't necessarily argue that movies need to subsume all tasks, right? I think they can be powerful for kind of discovery science or like more data driven, like okay, I guess there's sort of multiple answers to this point that I could give, but yeah, so I think you can also use like quite short clips, right? So that's like a way, but then you're kind of starting to approach this more traditional task-based, trial-based structure, and then at that point it's kind of like, what's the point? Like I do think I do think there's something to using slightly longer clips, like, I don't know, yeah, in the 5 to 10 minute range, let's say, or even beyond, that does let your participant kind of immerse themselves in this narrative that could then make that moment ultimately more powerful and ultimately more similar to what we might experience in our own lives, right? So like, I do think, and I hope I'm clear on, I mean, I do think it's important and good to kind of use movies in conjunction with maybe a more stripped down, controlled, trial-based paradigm. And hopefully there's some similarities between, for example, being like betrayed in a gambling game versus like watching a betrayal on screen, but that's an empirical question of like, you know, how much similarity is there between those two things. But if we use movies as a tool to get participants really kind of like invested in the story, I think that does lead to more powerful signals, and perhaps signals that are more akin to what we might experience in our own lives. I think the benefit of movies also is that, yes, it's nice and it's good to sort of go in with a hypothesis. And obviously I alluded to this when talking about picking a movie, is like ideally you have some sense of, you know, the effect you want the movie to have on your participants. And then you can model particular things about the movie that might induce or relate to that effect. But the nice thing about movies is they also give you a chance to kind of let the data tell you what's important, in the sense that if you see like a strong spike in correlated activity at a particular moment that you didn't anticipate, you know, doing that, you can kind of go back to the movie and say, well, what was happening at that point? Like, is there something here that I wasn't thinking to model, but maybe is important, or is a feature that the brain is leveraging or reacting to? Of course, then you probably have more work to do to then kind of build another context in which you are directly manipulating that thing, so that it's not just a big like reverse inference or storytelling game. But I do think movies give you that potential of letting the brain tell you what it cares about, as opposed to you as the experimenter kind of building a task in a way that you know exactly what you want to model. And again, that's very powerful. Like, I think we need both of these approaches. But I think that the benefit of movies over these more controlled tasks is it gives you a certain set of extra degrees of freedom, so to speak, such that you can go in with something that you might want to model and something that you might want to test, but it also leaves room for new moments or new features to present themselves that you can get from looking at the brain activity that you wouldn't have necessarily hypothesized in advance, and therefore you might not have baked into your controlled task. Yeah,
Definitely. Yeah, I mean, to some extent, I guess all of my questions kind of revolve, or are supposed to revolve, around the question of kind of, for what kind of question or type of science are movies more useful, and when is something else more useful?
I think, just to make one more point on this, again, like I don't think movies should subsume like all of fMRI research. I do think like one point that I've been trying to advance, like one place where I think they should play more of a role than they do right now is in these large scale consortium studies. So things like the Human Connectome Project, or even like UK Biobank, or the ABCD study. Like, these are really powerful kind of population imaging efforts where people are going to huge efforts and spending a lot of money to like acquire a lot of data on a lot of individuals. And that can be very powerful. But I think, in my view, it's a little bit unfortunate that a lot of the paradigms that get included in these studies are kind of these more traditional tasks that maybe, I mean, we've gotten a lot of mileage out of those, but there's also a lot of evidence now that those are not actually the best for revealing stable and meaningful interindividual differences. And those types of studies, sort of by definition, are kind of trying to do discovery science, right? It's like saying, let's collect a lot of data on a lot of people, and then we can kind of mine that in different ways. And it's not like you're going into one of those studies with a very specific hypothesis that you want to test. The idea is just to get a lot of data and kind of mine it in different ways. And so I think movies for that application could be a more powerful alternative to resting state, because you can do all the same things that you can do with resting state with movies, but you can also do all of these additional things where you can leverage the fact that you have that known time course to understand, you know, why different brains are sort of reacting, or maybe not why, but like sort of what the reactivity of different brains looks like to different features in the movie. And you can just kind of analyze that data much more flexibly, given that you have this ground truth time course of what was happening on screen. So I think that's one place where maybe they're underutilized relative to resting state, for example, and should maybe get more airtime in the scanner. But again, I think they're most powerful probably in conjunction with more targeted and kind of hypothesis-driven tasks to see if we can get a handle on the processes that we think we're studying in both of these contexts, to see how much they relate across contexts.
Yeah, so as I told you before we started recording, I interviewed Peter Bandettini a few weeks or months ago, and after we stopped recording, I mentioned that I was going to interview you, even though at the time I somehow missed that both of you that you'd worked for him or with him. And then when he said, that you worked in his lab for a few years, I was just curious whether there's anything he thought I should ask you about. And he gave some very good questions that all went way above my head and were way too technical about fMRI analysis. So I'm not going to ask any of those. But he did mention something I thought was very interesting, just as a kind of almost an aside, because he basically said you were the best writer. Basically, he's known as a collaborator. He basically said that, something to the effect of, usually you'd send him a manuscript for comments and he'd just say like, yeah, looks good. You want to submit it? And so I was very curious about that because writing is something that, let's say, I want to be good at. Yeah, it's a very broad question, but kind of how do you do that?
Well, first of all, that's a really nice compliment and vote of confidence from Peter. So I appreciate that. I've always loved to read and write. I've always been kind of inclined towards literature and journalism and stuff like that. So I do think that helped. And I actually think scientific writing should be less dissimilar to good journalism or even good fiction than it is. Yeah, I think there's a certain amount of conventions that we have to follow and that are useful, but I also think it's useful to kind of draw inspiration from other types of expository writing, like long form journalism, kind of see how people build up arguments in long form articles like that. It's not going to be directly applicable to the structure of a scientific paper, but I think at the end of the day, a good paper or a good grant kind of tells a story, right? It's a bit of a cliche, but it is for a reason. And, you have to keep your reader in mind, and someone's out there reading this and try to make it pleasant for them to read. I think that's your ideas will have a much bigger impact that way. And as to how to do that, I spend a lot of time on my writing. It's not something that comes quickly, but I think I like the metaphor. Again, this is probably overused, but writing is more like sculpting than painting, in the sense that, like, what can you subtract? Like, what can you take away to make it better? So you often start with just kind of like these messy drafts where you're just getting all the ideas on the paper, and then it's kind of like, okay, how can you streamline this? What can you cut? And like at every level, so it's like within a given sentence, can you like cut a few words, or is there a single word that can do what you're doing with a 3 or 4 word phrase, right? And even those small changes, those kind of add up and they make it easier and just more pleasant for people to read. And then also on a larger scale, like, why is this paragraph here? Like, what is it doing? Is it just here because you want to show off that you know this literature, but it's not actually super central to the argument that you're trying to make? And just kind of taking a very critical eye to every word, every sentence, every paragraph that's on the page, trying to make sure it's doing work and not just there as kind of a, oh, here's another sentence or here's another thing I could bring in. And it's hard. I think I sometimes go back to like my writing from a couple of years ago, and even that I'm sort of dissatisfied with it. But I really think it's writing with your reader in mind and just trying to be as concise as possible in what you're trying to say, and varying your sentence structure is sort of a classic piece of advice, but it can really help having some shorter sentences in there. But it's a labor of love, and I'm probably too much of a perfectionist with it, and it's never going to be perfect. And but I think it's a really important part of what we do is communicating it in written form. So I appreciate that Peter said that.
Yeah, I really like the point where you said that if one word can do the job of 4 words, something like that, because that's something that I do all the time, basically, even rewriting. It's just like, where am I spending, where's someone spending mentally 5 seconds on this when they could be spending 1 second on it? And I feel like it's something I do all the time, but it's never something I don't think I would have come up with it. But when you mentioned it, I was like, oh yeah, that is a big part of clarity, through just eliminating the stuff that doesn't need to be there or can be said shorter. Yeah. But I guess, I mean, did you do lots of writing and other things before? I mean, I guess you mentioned a bit of journalism or at MIT, the kind of press releases, that kind of stuff. Do you think that helps by just having kind of a contrast to what it isn't and to just knowing more specifically what you're trying to do with this research paper? Or does it just help because you've just thought more about language and how that works? And
I think it's helpful to have had practice and experience with other types of writing beyond science writing. I mean, I think most people that start a PhD program have probably written things in their lives, like, I don't know, I guess like research papers for school or other essays and stuff. So people will sort of come in with varying degrees of experience with that. I mean, I did always like, I was like growing up, I would like write little stories as a kid and illustrate them. And so I guess I've always been sort of attracted to the literary arts, so to speak. But I definitely think it's something that can be developed and cultivated, like even once you're in the field, and just kind of forcing like something I tell my students is when you're reading papers, which hopefully you are, sort of actually reading from start to finish, not all papers, some papers you will skim, some papers you will read in more depth. But when you find a paper that you think is really well written, just kind of file it away. And it doesn't even have to necessarily be for the content of the paper. But if you find a paper that's actually a joy to read as opposed to a bit of a chore to read, file that away somewhere and come back to it and see kind of, how did the authors craft that just on a very kind of craft over content. So like if you just break down the introduction into like what each paragraph is doing and also the style of language that they're using, can you to just start to deduce, what makes some papers more pleasurable to read than other papers, and use that when you're crafting your own papers to kind of do your readers a favor and make it interesting for them. And people will have different styles. Like I don't think there is one formula, but I think if you can kind of train yourself to start recognizing it when you see it, and you kind of compile some examples for yourself, you can kind of start to extract some principles of what you think makes a good paper, and then try to echo that in your own writing.
Yeah, and just briefly on the topic of papers that are joy to read, I guess I'll recapitulate that question now, because the first of my recurring questions is, what's a book or paper that more people should read? It can be famous, non-famous, old or new. I mean, I don't know whether you had whether this was already going to be your answer, but I'd also be curious if you have a paper that is a joy to read.
That's a good question. I probably should have been more prepared for. I mean, the paper that actually immediately came to mind when you told me you were going to pose this question over email is this paper from 2018. I actually have it right here so I don't butcher the title, but it's called The Reliability Paradox: Why Robust Cognitive Tasks Do Not Produce Reliable Individual Differences. The first author is Craig Hedge. It's published in 2018 in Behavioral Research. I love this paper. So this paper is actually very elegantly making the point from both a theoretical and an empirical perspective. I sort of vaguely alluded to this, I think, earlier in the conversation, but it's basically making the point that the sort of bread and butter tasks of cognitive psychology became that way, became bread and butter because they produce very reliable effects in basically almost every subject that you test. But that very feature makes them poorly suited for discovering meaningful individual differences. So if you have a task that is just very reliable in the sense that it kind of produces very stable behavioral effects, both within and across people, that task is well suited to understanding something generally about how humans or human brains perform that task, but it's not well suited to drawing out these meaningful differences across people, because by some of
the whole point is
I mean, some of the traditional definitions of reliability is like, well, you want to see similar effects in every subject that you run, right? And so that was true in like the earlier days of experimental psychology. It was also true in fMRI, right? Like a lot of the early papers were like, well, like you want to see motor cortex lighting up in every individual that you test, or at least most individuals that you test, and that's how you can kind of trust Like I think the original fusiform face area paper out of Nancy Canrisher's lab, like they were actually showing some individual subject data. They were sort of presenting results, and like, they discovered the fusiform in 9 out of the 12 people they tested. I can't remember the exact ratio, but it was like, that's a strength, when you're trying to prove something general about the population. However, it's a weakness when the thing that you actually care about is what is meaningfully different between people. And so you're kind of shooting yourself in the foot by using these like tried and true paradigms when you're trying to go after this variability across people. And so we kind of need to take a step back and think about designing new tasks and new paradigms that don't necessarily produce the same effect in everybody, but rather produce different effects in everybody in a way that's still sort of stable and reliable within each person, right? And that's challenging, but I think that's the way that we need to move towards if we're going to get traction on some of these individual difference questions. And so this paper, the reliability paradox, just kind of lays out the whole principle behind this in a very, I think, clear and elegant way, just like why it kind of has to be true theoretically. And then they also do some empirical analyses of some sort of classically used tasks like flanker task, Stroop task, stop signal task, and they kind of show that, the very feature that makes these tasks desirable for sort of like general cognitive psychology research makes them undesirable for individual difference research. So that's probably been my favorite paper in the last, several years, and I cite it all the time. And I think it's quite well written as well.
Nice. Second recurring question is something you wish you'd learned sooner. This can be from your work life, from your private life. I don't really care. Just something that you think that your life would have maybe been a little bit better if you'd figured that out a little bit sooner, and maybe how you figured it out or what you did about it.
Yeah, I think actually maybe this gets back to a second. I have a book recommendation. I actually just finished this book over the weekend, so this one is quite fresh. But there's a book called, I think it's called 4,000 Weeks, Time Management for Mortals, and it's by Oliver Burkeman. It's kind of this like existential self-help book, basically making the point that, we can sort of bend over backwards and try to be these productivity machines and fit everything in at work and at home, and just, sort of kind of just try to over optimize and maximize our time. But at the end of the day, we all have finite time here on this earth, and we're never, to the extent that we get more done, there's just going to be more to do, and it's kind of this never ending cycle. And so I'm a parent now, and I'm also a PI, and I also like to sometimes like do things that are not work, and it's challenging. And so I think the ideas in that book, I mean, it's not the first time that I had encountered some of them, but I think he lays it out in a particularly kind of engaging, compelling, often funny, often kind of tongue-in-cheek way. And I guess the point that I'm trying to make is like I wish that maybe I had been earlier on like a little less consumed with like, sorry, let me back up. This is like a hard thing to answer. I think one thing that's challenging about a career in academia is you kind of being your own boss sounds great, and it often is, but it's challenging in the sense of you have to kind of really manage yourself, and you have to do a lot of work. I often call it meta work. It's like work about work. So it's like you're thinking about not only work, but like, if you're doing enough work, am I doing too much? Am I doing too little? Am I focusing on the right things? It's like you have this not infinite, but you have a lot of flexibility and a lot of freedom as an academic to sort of work on things that you want to work on and choose, which activities you're going to do and which things you're going to say yes to. And that's great. But it also, it creates this level of sort of extra management that you have to do to try to understand, if you're actually using your time in the ways that you want to be, and you don't have a boss telling you like, prioritize this, not that, etc. And so I think that book really, crystallized a lot of the feelings that maybe I was having and maybe other folks have too, of like, it's just sort of getting obsessed with how you're using your time in any given day and whether you're using it optimally and not just at work, but in your personal life as well. And so I guess I wish that maybe earlier I had recognized this a bit better and recognized that there's always going to be more to do, and you're kind of always going to feel like there's infinite other things that you could be doing, but the best that you can do is just, pick the few things that are important to you at any one time, in terms of work, but also in terms of personal life, and just kind of focus on those things and recognize that life has chapters, right? And just because I'm not running marathons right now doesn't mean I can't do it later, or like something like that. And like to everything, its own season, and just to kind of stress out a little bit less about like the productivity geek culture and just like maximizing every moment.
Yeah, I mean that book, you're not the first to mention it.
Really?
I think Rachel Bader mentioned it.
Oh, cool.
And yeah, I also read it, I think a year ago or something, and I want to reread it again because it's reread it again. Yeah, I want to reread it. Speaking about when you can cut words. Yeah, because it does have, I agree, as you said, this kind of mixture of putting things in perspective, being weirdly calming at the same time as kind of telling you things you know but don't really want to hear. Totally. So yeah, well, I'll, with all the other stuff we mentioned, I'll put a link to that in the description. I would definitely recommend it. Final question: any advice for PhD students or postdocs, so people like me who are kind of at that transitory period between, finishing a PhD to having just started a postdoc? Yeah, I think specifically with respect to that period when you're kind of moving between PhD and postdoc, I think that's a really fun time. Maybe I'm not the only PI to have said this. I mean, I think when you're coming out of your PhD, you have a lot of skills and you have a good handle on the landscape of the field. And so when you're starting your postdoc, you're really kind of able to just go full steam ahead, and yeah, you already kind of have the skills on board and you can start to, make more rapid progress. At the same time, postdoc can be stressful because it's typically supposed to be shorter than PhD, and then you're worrying about the next step basically almost as soon as you start your postdoc. So that part is hard. But I would say like one thing that I think it's really nice and important to do as a postdoc is, and I didn't ask you exactly where you're planning to postdoc and how that relates to what you did in your PhD, but I do think postdoc, like, you have a lot of independence, but it's also nice in that it's kind of your last chance to get that direct training or mentoring from somebody. And I think, yeah, like you want to establish independence, but you also want to take advantage of that, being basically the last time in your life that you're going to have this like formal mentor who is in charge of you and like by definition needs to care about you and what you're doing. And just to kind of like lean into that and make sure like you're taking that advice on board and just kind of like maximizing that opportunity. I think postdoc is also a nice time to force yourself to get some experience. And maybe this is already what you're going to do. I think this is what many people try to do, but it's like you kind of want to add something to your toolkit that's a bit different than what you did as a PhD student, because that's going to make you a stronger scientist moving forward. Like I think actually when I joined Peter's lab was fantastic. And in many ways, Peter Bandatini was similar to my PhD mentor, Todd Constable, in that he's an MR physicist and he runs sort of a methods focused lab, but with very strong neuroscience applications. And so I actually think it would have been easy for me, honestly, in Peter's lab to just kind of do a lot of the same stuff that I'd been doing as a PhD student. And I think that's very tempting for folks at that stage, because it's like, as I just said, you have these skills and you kind of hit the ground running and you can start to crank out projects and you kind of like be really productive. And there's a seductiveness to that, which makes a lot of sense, because our field really values productivity and fast. But at the same time, I think like one thing I did as a postdoc almost by accident, but I think it was actually great, was like I kind of ended up doing this whole side project on layer specific fMRI, which is not something that I had done as a PhD student. It kind of gave me that exposure to a new set of methods and a new way of thinking about things. I think in many ways, like if you're planning to join a lab that's more different from what you did as a PhD student, this will come more naturally. But I think it can be hard to kind of slow yourself down and kind of force yourself to learn a new set of skills, because you do have all of this momentum propelling you in like whatever direction you're coming out from your PhD with. But I think that, yeah, kind of like forcing yourself to do something a bit different, where there's going to be more of a learning curve for you, and it might take a bit longer to like for that project to come to fruition, but it can be a really great thing to do to set yourself up to be in your own independent career, because now you kind of have this extra dimension that you can bring to problems that's, going to make you sort of more unique from what either your grad or your postdoc lab are doing. And it kind of, you kind of become this, you're like more than the sum of your parts almost, because you can kind of see connections and do stuff in a way that if you had just kind of stayed in the narrow lane,
that you'd acquired in your PhD. Like, sure, maybe you'd have published a couple more papers, but in the grand scheme of things, I think postdoc is a nice phase to kind of force yourself to do something a little bit outside your comfort zone, because you do kind of have that protected time and space to do that a little bit. Okay,
great. So learn something new by taking advantage that someone, for the last time in your life, is contractually obliged to care about you.
Yep.
Okay. Great. Well then, thank you very much.
It's a lot of fun. I hope it made some sense. So thanks for having me.
I think so. Thank you.
Yeah.